Okay. Hi, everybody. Welcome to the Broad MIT series in chemical biology and the first seminar of the spring series. So I'm delighted to introduce Professor Wilfred van der Donk. He's here from University of Illinois, Urbana-Champaign, to tell us about his terrific science this hour. Just as a little introduction, Wilfred began his education in the Netherlands and came to the US, where he did his PhD at Rice University, and then came to MIT for a postdoc with Joanne Stubbe, so welcome back yet again. And then began his career at University of Illinois, where he rose through the ranks and is currently the Richard E. Heckert Endowed Chair in Chemistry and also an investigator of the Howard Hughes Medical Institute since 2008. So Wilfred has received many awards. I'll just mention two of the most recent. In 2021, he was elected into the National Academy of Sciences and is the 2024 recipient of the Bader Award from the American Chemical Society. This is an award for excellence in either bioorganic or bioinorganic chemistry. His research program really is at the intersection of chemistry and biology, focused on discovery and fundamental understanding of biosynthetic pathways, enzymology, and new natural products. So Wilfred, welcome back. Delighted for your talk.
Great. Thanks, Liz. Can you hear me? Okay, yeah, is it working out? Thanks for the kind introduction. I would also like to thank the faculty of the Broad and the MIT Chemistry Department for the invitation. I was really looking forward to this. Ahmed invited me about 2 years ago, I think, but I couldn't at the time make it for various reasons. And so I've really been looking forward to my visit, and it has been as inspirational as I anticipated, so it's been really great. The title that I gave, again, a long time ago, was Genome Mining for New Natural Products, as well as New Chemistry. I will mostly focus on the latter part, so genome mining for new chemistry. And so why would we genome mine for new chemistry? Well, of course, there's the inherent interest in the chemistry that takes place in nature. But also, over the last 10, 15 years, there has been another very good reason to look for new enzymatic reactions, and that is the real embrace that the industry has shown to using biocatalysis in the biosynthesis and chemoenzymatic synthesis of pharmaceuticals. And so I think a real watershed moment was this 2010 paper from scientists at Merck and Codexis, where they showed they could take an enzyme and through directed evolution use that enzyme to set that stereo center here in this then clinical candidate, Januvia. And so that trend has continued, and so in process chemistry, using enzymes has become a very popular way to reduce waste and increase the efficiency of making pharmaceuticals. So a few more examples are shown here. These are molecules that were discovered by medicinal chemistry using purely synthetic chemistry. But once they went into clinical development and hundreds of kilograms were needed, chemoenzymatic routes turned out to be really the most efficient. And so if you're a process chemist in industry and you are given a molecule that is already set, the molecule has gone through the development and now it needs to be made in large scale for the clinical trials. If you want to make it chemically, you have 100 years of methodology development to probably come up with a dozen or more routes to make the molecule. But if you want to use enzymes, that is not the case. Actually, the number of enzymes that are available that allow you to use enzymatic steps in a chemoenzymatic pathway is very, very limited. And indeed, all of these examples use enzymes that did known chemistry that had been studied previously. The directed evolution essentially allowed them to take substrates that they naturally do not take. And so if you read these papers, there's often statements like this. This is taken, I forget from which one. It's a quote that for this to continue, we really need to discover new enzymatic transformations, such that hopefully at some point for the biocatalysis, you also have multiple retrosynthetic routes by which you could try and make a molecule very efficiently. And so that started making us think as to how do you discover fundamentally new enzymatic reactions. And so there's a number of ways I think you can do it. You can do it the way Francis Arnold and people coming out of the Arnold lab have done, where you essentially have enzymes do reactions for which there is no natural counterpart. And that has been very successful. Another way to do it is to say, okay, there's still thousands upon thousands of unknown proteins, genes that are annotated in the databases as hypothetical proteins or domain of unknown function. And some of those might also be doing interesting chemistry. And especially in the realm of natural products, there's amazing structures. And of course, these need to be biosynthesized, and there might be really interesting chemistry that is hiding there. And so we started then a program where we wanted to intentionally look for enzymes that we didn't know what they might be doing. Now, that's easier said than done. My colleague, John Gerld, for about 10 years, ran this enzyme function initiative, and the whole goal there was to assign functions to proteins of unknown function. And he would tell you, and his colleagues would tell you, that the hardest thing is to figure out what the substrate is going to be of the enzyme. If you don't know what the substrate is, you cannot really know what the enzyme is doing. And that's where we thought we had a really unique angle, because we had been working on the biosynthesis of a class of natural products that are called RIPs. These are ribosomally synthesized peptides. They're gene encoded. And then because these structures are limited to the 20 natural amino acids, what nature does is a large number of post-translational modifications on part of that peptide that we call the core peptide. I'll show you a few structures. These are often macrocyclizations, but also all kinds of other chemical transformations. A leader peptide that is important to guide these enzymes is then cut off, and then you have the natural product class that are now referred to as RIPs. And so RIPs were long believed to be a very small group of natural products, but with all of these genomes that have come out, we now know that it's one of the largest classes of natural products on Earth, and that is because they're made by all domains of life, in all domains of life. So eukarya, archaea, bacteria, and among the bacteria, almost all phyla make RIPs. So it's not limited to, say, actinomycetes or cyanobacteria. And so if we think about this pathway, it's really perfectly set up for a number of different things. One is to do genome mining for new natural products. I'm not really going to talk about that today, because everything here is gene encoded, and hence you can turn on the computer, look for biosynthetic gene clusters that are making molecules that you've never seen, and you can then transport these pathways into heterologous hosts like E. coli, yeast, and so on. But for this talk, it also is a great way to discover new chemistry. Suppose that this is a hypothetical gene cluster, and that the computer says about the yellow and the green enzymes that those are domains of unknown functions, or enzymes that are hypothetical proteins. Because the little gene there encodes for the precursor peptide, we have a direct route into assigning the function of these enzymes. We know that one of the genes in that cluster is making an enzyme that does the first reaction on that peptide. The product of that is going to be taken by the next enzyme. And so by co-expressing different combinations of enzymes, we can walk towards the functions of these hypothetical proteins. So in our lab, we love biosynthetic gene clusters that have a lot of domains of unknown functions or hypothetical proteins, because that allows us to find new chemistry, potentially. And so let me first show you a few structures to have an idea as to what are these post-translational modifications that are introduced. And so here are just 5 of what are now more than 50 classes of natural products. In the thiopeptides, you see that there's pyridines and thiazoles. And one thing that we see often is that the amides, that of course are a vulnerability for peptides with respect to proteolytic degradation, that often these are converted into other structures, such as these thiazoles and oxazoles, or thioamides, or methylated amides, or N-to-C cyclization, side chain cyclization. As I mentioned, many of these have macrocyclic structures to impose a conformational constraint that makes these molecules active. And so the first example where we were able to use this approach to assign function to proteins of unknown function is a little dated now, but it sets the rest of the story. My two postdocs, Chi Ting and Michael Fung, decided to work on this biosynthetic gene cluster from Pseudomonas syringae, that's a plant pathogen. And we had a number of questions. We didn't know what the enzyme encoded by this dark blue gene might be doing. And we didn't know what this protein encoded by the gray gene H was doing, but we knew it was a member of the domain of unknown function 692. And we also knew that there was a really large protein family for which no functions had been determined previously. So using this combinatorial co-expression in E. coli, Chi and Michael were able to show that the first step is actually catalyzed by that blue, the enzyme encoded by the blue gene. And it does a reaction that they hadn't really anticipated. So the enzyme adds cysteine from cysteine tRNA to the end of the little peptide that is encoded there. So essentially the same chemistry that the ribosome would do, but without a template. No mRNA is present there. So we didn't really know what to make of that, but it became clear that it must be on pathway, because after the cysteine is added, now these DUF692 enzyme, and it needs this partner protein, that's why we also have colored that in gray, and it does a reaction that we have really never seen before. So the enzyme excises the beta carbon as formate. We can show that using carbon-13, and then reattaches the sulfur to the alpha carbon with retention of configuration. Then the steps after that are a little bit more traditional. So the two yellow proteins also collaborate. The E enzyme takes SAM and turns it into carboxy-STAM here. And then the F enzyme then transfers that carboxy-methyl group to that sulfur to make now an analog of glutamate. We call it thioglutamate. And then there's a protease in the gene cluster that then cuts off what we have just made. We regenerate the precursor peptide. It can now go around and around and around. So we have catalytic use of the precursor peptide. It's a scaffold on which a natural product is being bound. So when we had discovered this, we wondered, okay, how often do we find these type of pathways? So I'm going to tell you about other examples of these enzymes, which we now call PEARLs for peptide aminoacyl tRNA ligases. I'm going to start out by telling you more about this DUF692 enzyme family, of which there's about 16,000 in the current genomes. And so if we make a so-called sequence similarity network, using the tools that have been developed at the Enzyme Function Institute, something that was started with Patsy Babbitt, these types of renditions of sequence similarity networks, and then John Gurlt really has popularized them. So every dot that you're looking at is an enzyme. If the dots are close together, then there's high enzyme similarity. If the dots are further away from one another, the sequence similarity is lower. And then you can set certain cutoffs by which the enzyme families are put into different groups. So at this particular cutoff, the assumption is that these blue enzymes are all isofunctional. They're going to do the same reaction, but a reaction that is different from, say, the red ones or the pink ones or the orange ones. So I just showed you what the green ones are doing here. And actually, as we were working on this, another paper came out from Amy Rosenzweig's lab that worked on the pink ones, and she showed that that enzyme family does, again, a remarkable chemical transformation. I challenge you to draw a mechanism to turn a cysteine into an oxazolone thioamide there. So now we had two enzymes, two really unusual reactions, but 16,000 others. And so we wanted to see what else was in this enzyme family. And so before I show you some of other examples of groups on this sequence similarity network, let me first show you a crystal structure of one of the pink ones that was solved in China in Chengdu by Wei Cheng and his co-workers. And so in blue, you see the protein. This is the DUF692. In pink is its partner protein that binds the substrate. And in yellow, you see the substrate. And so when we zoom into the active site, we see that this is an unusual enzyme family because it's a trinuclear iron-dependent enzyme. And so if you look at all 16,000 of these proteins, the ligands for 3 irons are almost entirely conserved. And so that was very interesting. The cysteine of the substrate, you can see, makes a bonding contact to one of those irons. And so based on the structure and the reaction that our enzyme catalyzes, we can then propose a mechanism. And so that is shown here. So I'll first go over the mechanism, and then I'll tell you what our support is for this mechanism. And so we know that we have 3 irons in our enzyme also. We have also crystal structures with Wei-Cheng in a collaboration that also contain 3 irons. We know that at least one of them needs to be reduced. We don't know if they all need to be reduced. But if they're all oxidized, we have no activity. We know we need oxygen. The reaction doesn't go without oxygen. And so we believe that we first make a ferric superoxo species. And we think that that does a hydrogen atom abstraction of this activated position here at the beta carbon. That would generate this radical, and you can actually draw a number of different mechanisms to go from this radical to this thiohemi-aminal. And so this is just one way by which you can draw this. This also can go by other steps. If that happens, then we make an iron 4-oxo species next to this alcohol. And we believe that that might do an OH oxidation. Of course, an OH bond is very strong, but we're also making a new OH bond, so thermodynamically it should be okay. And in another enzyme system that is a mononuclear iron-dependent enzyme, in collaboration with Marty Bollinger and Karsten Krebs, I think we have very strong evidence that a ferryle can oxidize an OH bond to make an oxygen-centered radical. What probably pulls the whole system forward is that if you do generate that radical, very rapidly you will do a beta scission to make a much more stabilized radical, a glycyl-type radical. That could potentially then do a rebound to make the observed carbon-sulfur bond, and it could then also lead to a hydrolysis of this thioester to give the 2 products. So the support that we have for this is that if we put deuterium on the alpha carbon, we see no change. The deuterium is retained, so we don't believe that there's any chemistry that happens on the alpha carbon. If we put deuteriums on the beta carbons, one ends up in the product, the other is lost into solvent, as we would expect. Perhaps the most telling is that if we do the reaction with O18 labeled oxygen, only one O18 ends up in the product, suggesting that the second oxygen comes from solvent, comes from water. And so, of course, that doesn't prove this mechanism. And we would love to try and trap some of these intermediates, as we did do for that other enzyme in this paper. Thus far, that hasn't really been successful. And so we're still working with Marty and Karsten to see if we can get more direct spectroscopic evidence for this particular mechanism. Now, a few points. The first thing I want you to remember that here we have a carbon that is at the thioaldehyde oxidation state. And that's because later on I will show it again, but I will not show these first steps leading to that intermediate. Okay, so back to the sequence similarity network then. Can we discover other reactions in this enzyme family? And so I'm first going to tell you about the orange family here. It seems like I'm starting to run out of battery. I'll change it in a second. And so Richard Ayikwe, he was a postdoc in the lab, started working on that. He picked one gene cluster from Chrysiobacterium. It contains a precursor peptide that you can see the sequence of. All orange proteins from the previous slide, they have sequences of precursor peptides that always end in the last 7 amino acids that are identical, that are bolded here. And so what Richard was able to show, that if you then put this into E. coli, you do co-expressions. If you co-express the substrate with the DUF692 enzyme in red and this partner protein in purple, you get an addition of 10 Daltons. And so the question is, what is 10 Daltons? We can treat the peptide with a protease that cleaves right here at this lysine. And from that, we know that the 10 Daltons are added at the C-terminus of the peptide. And so at that moment then, we decided we need to do NMR. That's really the only way by which we were able to determine what exactly the change in mass is due to. And it turns out that the change of 10 Daltons is a very complicated change because it's removal of 4 hydrogens and then addition of a methyl group. So it's -4 and then +14, and that gives you the net change of +10. I must have put them in the wrong way. I had 50% chance there. And so the, let me change them around. Because I can do it the old-fashioned way. So that's the reaction that the enzyme catalyzes. As you can see, a lot of different things are happening here. And so the enzyme makes two rings, so that's loss of 2 hydrogens to make this ring, a loss of 2 hydrogens to make that ring, so that's a loss of 4 hydrogens. And then that is the addition of the methyl group. So to orient you, this sulfur makes a new bond to this carbon. This sulfur migrates to that carbon. Instead of breaking this bond that I showed you previously, in this case, we're going to break this bond, and this nitrogen picks up that carbonyl to make this hydantoin structure. And then the sulfur that migrates to the alpha carbon is then methylated, and we have shown that that comes from acid-anisole methionine. We don't know the absolute stereochemistry yet here at these 2 positions. And so we have a lot of NMR data that supports the structure, and we've done quite a bit of labeling data. So we know that these alpha carbons end up at these positions. More tellingly, if we label the carbonyl of the cysteines, we can show that this carbonyl ends up here at the hydantoin. So certainly a very unusual reaction. Not sure that this is going to be a useful reaction, but certainly new chemistry. And so we have renamed the DUF692 family then as the multinuclear iron-dependent oxidation enzymes. Why not trinuclear? Well, because there's some disagreement in the literature between 2 groups on not our enzymes, but another enzyme, whether the active enzyme is trinuclear or dinuclear. And so for now, we just want to make sure that we don't bias that argument. And so then going back to the potential mechanism, we don't have any evidence for this mechanism. But as you will see, there will be similarities and commonalities between the previous mechanism. And I start out with that first intermediate that I reminded you to remember, a thioaldehyde and then a ferric superoxide. So in the previous mechanism, we had this hydroxyl attack it to do a hydration. In this case, we used the nitrogen of the next amide, the next amino acid, to attack the thioaldehyde. That would give us this intermediate beta-lactam. Then we believe that this nitrogen, which we, remember, need to pick up this carbonyl, could attack the beta-lactam. And then we would generate an alcohol that is now set up for the same type of chemistry, which is oxidation, beta scission, generation of a radical that now can form an episulfide. And then the second cysteine can open that up, and we can then methylate that sulfur. And that's why we really want to know the stereochemistry and are working hard towards understanding the stereochemistry, because there's clear predictions here about what the stereochemistry ought to be based on this mechanism. So whether this is correct, we don't know, but we do know that it at least uses similar type of chemistry as the previous mechanism. And Amy, in her mechanisms for the cyanobactin biosynthetic enzymes, also has very similar mechanisms with respect to what might be happening. So I've shown you now 3 reactions, all on cysteines and all unprecedented and not predictable. I don't want to leave you with the idea that these enzymes only act on cysteines, because we have several examples where that's not the case. And so one example is shown here. This was the work of Din Nguyen, who is now here at MIT in Michael Laup's lab. He was a joint student with my colleague Doug Mitchell, and he worked on this cluster from Burkholderia thailandensis. And again, this combination of enzymes we have never seen before. We like that. And he was able to show that there's a B12 radical SEM enzyme that first methylates on the beta carbon of this tyrosine with the shown stereochemistry. And then a P450 takes the 2 tyrosines and it cross-links them, but it does so with the stereochemistry that I'm showing you there. So you get only 1 atropisomer, and we know that because we have a micro-ED structure of the product. And then the MNIO acts, as I already mentioned, not on a cysteine. In this case, it acts on a C-terminal aspartate and turns that into a beta keto amino acid, a structure that medicinal chemists have used for a long time to make inhibitors of proteases. And then the last step, this methyltransferase here, methylates at this position, and then was able to show that this is indeed a cysteine protease inhibitor. We do not know why the Burkhold area is making this molecule. But again, unprecedented chemistry. And the final example that I want to show you is actually from unpublished work for my current postdoc, Shekhar Padi. So he picked yet another gene cluster. In this case, again, a combination of biosynthetic enzymes that we have not seen before. There's about a few hundred examples of those. And this sequence similarity logo shows that this part of the substrate that is bolded here is fully conserved in all of these several hundred precursor peptides. And so what Shekhar was able to show is that in this case, the MNIO enzyme acts first. So all of these pathways are strictly ordered. In this case, nothing happens until the MNIO has done a reaction. And as you can see, the chemistry is again very different. In this case, it takes 2 phenylalanines and makes 2 orthotyrosines. And then after that, this arginase acts, and it takes this fully conserved arginine and turns it into an ornithine. And then another radical SEM enzyme does again macrocyclization to now make a different ring pattern. And again, we do not know why these organisms are making that. At the moment, we're mostly focused on discovering new enzymatic chemistry, as I said in the title. Of course, we're very interested also why these organisms are making all of these macrocyclic peptides. But for now, we have put that on the side, and we will go back to that later. And so I've shown you now 5 different reactions, all are unprecedented. And other labs have also contributed to these. And so Mo Sayed-Sayamdost, who actually was a student here with Joanne, he showed yet another reaction where this carbon-nitrogen bond in the backbone of a peptide is broken to make an amidated peptide. And the group of Jacob Dubrisson in France showed yet another reaction on cysteine, where cysteine is converted to this thiooxazole. So I don't know if any of these reactions will ever be useful for a directed evolution campaign to make structures. I do know that they make a lot of heterocycles, and of course heterocycles are quite frequently found in natural products, and whether it's an oxazolone or a hydantoin or thiooxazole, these are all structures that we haven't really seen in natural products. So let me return then to these Pearl enzymes, these enzymes that add an amino acid at the end of a peptide. At first glance, a silly reaction, because you could have encoded the cysteine on the DNA and the ribosome could have made this. But after we figured out this catalytic cycle, it makes a lot of sense, because you now have a catalytic peptide. You don't have to make a 50 amino acid peptide every single time you want to make a natural product. And so we have then started looking for other enzymes like these. And at last count, we have now 8 of them that all add different amino acids and all in an amino-acyl tRNA-dependent fashion. And I think when we're all going to be done with this, we're going to find cases of all 20 amino acids. I'm pretty sure that there's going to be enzymes out there that add any of the 20 natural amino acids to the end of peptides. They all make different natural products. The biosynthetic pathways diverge after the addition of the amino acid. But we think that this is quite interesting, given the real interest there is currently in the pharmaceutical industry in peptidic therapeutics. Because those can be made by solid-phase peptide synthesis, but maybe some steps also can be done using biocatalysis, and so then these enzymes might actually be quite useful. And so we have started to try to understand how they work. And so first, some isotopic labeling studies that were done by Zhenggang Zhang. He first put an O18 at the end of the peptide, was able to, that O18 is actually liberated as phosphate. The reaction requires ATP. And so we believe that we first get a phosphorylation to make an acyl phosphate activated carboxylate, that the nitrogen then of the amino-acyl tRNA attacks that to make the amide bond. And then doing the reaction in O18 labeled water showed that actually the activated ester that the ribosome uses to make proteins, our enzyme just hydrolyzes it. So it seems very wasteful. It seems a prime example the opportunistic manner of evolution, presumably all these organisms that use these type of enzymes, they benefit from the natural product that they're making. But they're certainly not making it in the energetically most efficient manner. And so we don't have crystal structures of these enzymes. So we don't really know how this all fits together. But then when AlphaFold 3 came out last year, my current postdoc, Jocelyn Ramos-Figueroa, put it all together. So protein, peptide substrate, ATP, and tRNA. And the prediction is rather stunning, I think, knowing that there are no examples of structural information on these. So AlphaFold places the 3' end of the tRNA, where the amino acid would be attached, right in the same vicinity where it places the C-terminus of the precursor peptide. And that C-terminal carboxylate is right next to the gamma phosphates of ATP. So it's exactly set up for the chemistry that we know the enzyme does, but that AlphaFold, of course, doesn't know. And so the phosphorylation, and then here is where the amino acid would be attached, ready to attack that carboxylate there. And so in 3 dimensions, it's a little bit better to see. And the other thing I want to point out, up here, it looks like there is a contact with the anticodon loop of the tRNA. And so it seems like there's 2-point interaction in order to make sure that the right amino acid gets put on, both based on the anticodon as well as based on the amino acid. So we're trying to investigate this right now. Again, my student Din Nguyen, and he's working together with Hiro Zuga, who has this FlexEnzyme technology that allows you to change the tRNA and the amino acid and put different amino acids on different tRNAs and vice versa to see whether that recognition is indeed real of the anticodon loop. Jocelyn has already made the mutations of all the conserved residues that AlphaFold predicts are important for recognition of ATP and of the aminoacyl tRNA. And indeed, if we mutate that tyrosine or that arginine that are fully conserved and that are making contacts to this, the 3' CCA arm that, of course, every tRNA has, we lose all of the activity. So we believe that these predictions are actually potentially quite correct. So if we assign functions to enzymes that only are involved in pearls, then I think it would be useful, but it would still be, involved in rib biosynthesis. That would be useful, but still somewhat limited. But fortunately, quite often, the enzymes that we study are also found in other contexts, sequence contexts, other gene clusters. And so I want to show you that in the context of these pearls, because we also find those in polyketide synthase gene clusters and also in non-ribosomal peptide synthetase gene clusters. And so then the question is, what do they do in that context? Sometimes they are attached to thioesterase domains, sometimes they are part of a complete module, and sometimes they're standalone pearls. And so we have started to investigate these as well then, and this is the work of UAU, a current student in the lab. So he picked a gene cluster from Stachybrandtia nassauensis. It contains an NRPS, a PKS, a thioesterase, a dehydrogenase, and then the pearl that we want to know what is it doing in this context. And so he first took the NRPS and the PKS, expressed it in the heterologous host Streptomyces albus, and by doing unbiased metabolomics was able to show that 2 new molecules are being made, compound 2 and compound 1. It turns out that compound 2 turns into compound 1 as you are doing the purification and the structural elucidation. And so compound 2 turns out to be a diarginal compound that has a urea in the middle and then an ethyl ketone at the end. And compound 1 is where this nitrogen reacts with that ketone to cyclize it and turn it into this structure. So then when we put in the pearl also, so co-express the pearl together with the NRPS and the PKS, we again get 2 compounds. And the difference between what I just showed you is that now 2 threonines have been added to the structure, suggesting that the pearl is adding a threonine. And at first glance, you might say, well, you have an NRPS. This is already making a peptide. Why would you need another enzyme to make another amide bond? And the answer lies in the fact that you have made a urea. So if you've made a urea, you have essentially inverted the directionality of the peptide. And it was already pointed out by Chris Wallace in this publication when he studied urea formation in non-ribosomal peptide synthesis. This is his proposed mechanism where the nitrogen can react with bicarbonate, and he shows that that is the source of the carbonyl in the urea. Then you can draw a number of different mechanisms. I will just describe one. If this oxygen attacks that carbonyl there, you would make an anhydride-like structure, and then you could have an O to N carbonyl shift in order to give you the urea structure. Now, if you think about this structure, this is activated to do more chemistry at this thioester. But at this other carboxylate, with assembly line chemistry, you cannot really do anything with it. And that explains that when we look at the dictionary of natural products and we look at all urea-containing molecules, they always have only a single amino acid on one side of the urea. And then on the other side, in black, is where further elaboration by PKS or non-ribosomal peptide synthesis has taken place. And that shows that the system cannot do chemistry here. Apparently, that if a natural product can be made that is beneficial to the organism by doing further chemistry here, a pearl is attracted into the biosynthetic pathway, because now you can phosphorylate here. We have shown that the threonine comes from 3NL tRNA. And so now you can also extend in the other direction. And so very curious, there's a few thousand examples of this in the databases. What other structures are being made? To finish the biosynthesis of this particular structure, the polyketide synthase then adds a methylmalonyl unit to this thioester here. Then the thioesterase cleaves this off the thiolation domain. And then the dehydrogenase domain that I haven't really talked to you about thus far, it actually desaturates this ethyl group to this alpha-beta unsaturated ketone. And that's why we left it out of our experiments. Initially, we had put it in, but this is so reactive that you constantly find other things that have added here, and that really made the study much more difficult. So we left that out in the studies that I just showed you. So far, I hopefully have convinced you that we can do genome mining for RIPs, and that I can also assign function then to these hypothetical proteins, that these pearls add amino acids to both ribosomal and non-ribosomal peptides, and that these MNIOs really are incredible catalysts that catalyze all kinds of different oxidative reactions. In the last few minutes, I would like to very briefly touch upon some other things that I normally talk about when I don't talk about genome mining for chemistry. And that is that these biosynthetic pathways are really versatile to do bioengineering, that you can do genome mining in an automated fashion. We've done this together with my colleague Huimin Zhao, who has these robots that drive up and down, do all of the molecular biology to refactor biosynthetic gene clusters into plasmids that we then put into E. coli to then produce the RIPs. And I want to start out with pointing out that these enzymes are really great to make libraries of polycyclic peptides, another popular new drug modality that is being studied a lot in the pharmaceutical companies. So this is just a summary slide showing all of the different RIPs that to date have been turned into libraries, like 10 to the 6 to 10 to the 8 variants of polycyclic peptides, because these enzymes are very forgiving with respect to the sequence of the core peptide. So we have done this for lentipeptides. We've done phage display of these compounds, yeast display. We've done reverse to hybrid experiments. We've done epitope graphing. Hirozugo and Yuki Goto have done mRNA display of these thiopeptides. Here at MIT, Chris Voigt has made libraries of these rentipeptides and then selected them for binding to certain target proteins. David Craig in Australia has done a lot of epitope grafting on these plant cyclotides. And Doug Mitchell and Konstantin Severanov have made libraries of lasopeptides and then selected those for binding to other proteins to disrupt protein-protein interactions. So I think this is a really tremendous space to make libraries of cyclic peptides by using enzymatic means, which means you can access very large libraries. With respect to bioengineering, I wanted to point out that if you do mRNA display or yeast display or phage display, typically you then look for binding with a purified protein that is maybe labeled or maybe fluorescently labeled, and you can find binders to all kinds of different proteins. But it would be really nice if you could do this in the context of human cells, where there might be protein-protein complexes, there might be glycosylations happening. And so can we do this in mammalian cells? Can we make these bacterial and fungal products in mammalian cells? And I've already pointed out that these syntheses are incredibly portable because all you need is a ribosome and 20 amino acids. And so indeed, we have recently shown that we can make these molecules in HEC293 cells. This is one example where this compound here was made, and you can see from the fragmentation and also from the stereochemistry and the bioactivity, we are making the same molecule that in this case an Enterococcus faecalis make in its own setting. And in this case, it's a compound from a bacillus strain, and again, we get the exact same outcome. But the nice thing is because these are peptidic, you can send the products to where you want to make it go. For instance, if we want the cyclic peptide to go to the nucleus, we can add the nuclear localization sequence. And in this, let's focus just on this here. You can see if we don't have the nuclear localization sequence, the product is in the cytoplasm. And if we add the nuclear localization sequence, now the product goes to the nucleus. These were transient transfections, and hence only 2 out of maybe 10 cells are transfected in this case. We can, of course, increase the transfection efficiency or do selections, which we didn't do in this particular case. We can also send these products to the cytoplasm, to the cytoplasmic membrane by, again, adding a cytoplasmic localization sequence. We can send them to the ER. And when we then take them out and see are they still fully processed, in all cases, for this particular example, the 8 dehydrations and the 3 cyclizations had still taken place. So I think there's potential here to start looking for disrupting protein-protein interaction and doing it in the native mammalian context rather than doing it by mRNA display or yeast display or some other in vitro method. And then to end, since I mentioned, I had originally in my title that I was going to tell you about genome mining for new natural products, and I haven't really told you anything about that. Let me just point out 2 ribs that come from our own microbiome and that are medically important. Neither of these were discovered by us, but both of them were made in E. coli by us, and we have been collaborating with the people that have been working on them. So this is a molecule that is called blottosin that was discovered by Eric Palmer and his co-workers now at the University of Chicago. And he shows in this paper that if you have in your microbiome this organism, Bladia producta, that makes this blottosin, that you are protected from establishment of vancomycin-resistant enterococci infection. So these can no longer establish in the gut area. And so we're working with Eric's lab to see if we can improve the activity of this molecule, either as a therapeutic or as a prophylactic. Now, not all ribs that are made in our gut are good, and one that is particularly bad is cytolysin. This is made by pathogenic Enterococcus faecalis. It was reported first in the 1930s that this was leading to death. And in 2014, I think it was, we determined the structure by making it in E. coli and then determining the structure of the compound. When we published that, we were contacted by a lot of different labs that asked, can you send us some? And one of those labs is Bernd Schnabel, an MD at UCSD. He sees infectious disease patients. And when he asked for the material, he said, all the patients that I see, they are going to die. So these are patient numbers that are infected by Enterococcus faecalis that can make cytolysin. I should point out that this is a specific form of liver disease. This is alcoholic hepatitis. And so there is no cure. The people that come into his office will die. And so after we sent him the cytolysin, he was able to show, and that's all his work, that it is indeed a cytolysin that is directly responsible for the destruction of the liver of these patients that eventually leads to their death. So we're now working on inhibition of the biosynthesis of cytolysin to see if that can lead to a potential treatment. So with that, let me thank the people that have done the work. You've seen their pictures. Let me tell you where they are now. Richard is at Merck. Chi-Ting is down the road at Brandeis. Michael is at Science Magazine. Jocelyn and Shekhar are still in my lab. Zengar is now at Shandong University. Din, as I mentioned, is here at MIT in Michael Laub's lab. Sarah is at Just Evotech. Yue is still in my lab. And we've had great collaborations with Doug Mitchell's lab. I want to thank our funders. I want to again thank Ahmed and Liz for the invitation and you for your attention. Thank you.
Great, thank you. So the floor is open for questions, and we're in a really big room. So there's one mic over there. If people on that side want to get up and go, and I can run it here or put it. And I guess in the spirit of this series, we'll ask for trainee questions first.
Question in the middle.
Where are you?
Do you guys ever look at induction of these biosynthetic gene clusters?
Yeah, we do, and that's a bit of a sore point. So we often buy the organism that we get the cluster from. Most of the genes, I didn't really say that, but all of the genes were E. coli optimized. They were synthetic. They came from Twist Biosciences. But then quite often when we have made it, we don't know what the activity is of the molecule, and we buy the organism and see if we can tweak it for production, because maybe we didn't make what the natural organism make. And I would say 90% of the times we cannot find conditions where the natural biosynthetic gene cluster is turned on. And we try all kinds of different things, stressing, adding antibiotics. There's a lot of things that people have developed. And this seems to be a general thing in the RIP space. So I've heard Jörn Peel at the ATH complain to me that these RIPs never actually are made. And so it is indeed something that is a problem, because we cannot always test. Clearly, the reactions that these enzymes carry out are the reactions they carry out. But what the function is of the natural products, we would like to know too. And then if we have made the wrong compound, or perhaps we're missing a step, or perhaps not all of the reactions were performed the right way, we would like to get the natural product, and most of the times we don't see it.
Go to the microphone over there. Yeah.
Hi, thank you for the great talk. And about the mechanistic studies of the multi-iron enzyme. So like you said, you're gonna do some spectroscopic studies. So are the intermediates, are the time scale, is it allowed to do like some freeze-quench studies, or?
Yeah, that's exactly how. So in the study on the non-heme mononuclear iron-dependent enzyme that I alluded to, we did both stop-flow studies in my lab, and then also freeze-quench studies in Mossbauer and EPR in Marty and Karsten's lab, to stitch together seeing the ferrule species and seeing that the decay of the ferrule species had both an isotope effect on the solvent and a substrate isotope effect. And I haven't shown you that enzyme, but that can really only be explained if it is the OH bond that is being activated. And so that's what we have been trying to do. Unfortunately, not all enzymes are equally well suited for these type of experiments. You need high concentrations. In some enzymes, you cannot get high concentrations. You need a certain window of the kinetics in order to be able to trap them. And so we are very hopeful in the first enzyme I showed you that breaks the alpha-beta carbon bond. The second enzyme is not well behaved that I showed you.
Thanks for the talk. How do you plan to inhibit the biosynthesis of the cytolysine? Is there one enzyme that is particularly easy to destroy?
All RIBs need to have the leader peptide removed. And without the leader peptide being removed, they are inactive. So that's one. The enzyme that makes the rings, I didn't show you this at all, but it has a mammalian lipid kinase fold. So my colleague Satish Nair was able to get a crystal structure of these enzymes. Closest structural homologue is actually mTOR. And clearly lots and lots of compounds out there already that inhibit mTOR that we are seeing if we can repurpose them to inhibit this bacterial kinase. But this bacterial kinase first phosphorylates and then eliminates the phosphate to put the double bond in to make a dihydroamino acid. So it will be quite different. We already know because we've published the crystal structure. The active site is different from, say, mTOR. So we're not worried about treating patients to try that cytolysine is no longer make, and then at the same time inhibiting mTOR and getting all kinds of other effects that we don't want. But at the moment, I don't have anything that looks particularly exciting, but that's what we're doing.
Hey, Wilfred, this is very interesting. The Pearl enzymes. So, do you see any evidence for codon bias, or do you think they accept all the different cognate tRNAs for?
Yeah, no, it's a great question. So we have one that adds leucine on, and so in bacteria there's 6 different ISO accepted tRNAs. All of them work, and so we don't. Yeah, we clearly need to do more. So if there is an anti-codon loop recognition, that tells you that it can only be to some of the nucleotides. But yes, we have not seen any. When we first discovered them, we thought that maybe this is related to secondary metabolism. Certain tRNAs are induced late in the life cycle of bacteria. So far, at least in vitro, we don't see any preference for one or the other.
But they have to accept the tRNA for that amino acid. If you give them a different tRNA, they won't work?
Correct. Yeah, but with Hirozuga, we can put the wrong amino acid on the right tRNA and ask, does it still work? And what if you put the right amino acid on the wrong tRNA? But those studies are ongoing, so I can't really tell you yet.
I like your orange T-shirts.
Yeah, they do double duty. It's the color of Illinois and it's the color of the Netherlands, and so we have to.
From total football to total science, right?
There you go.
I just had a follow-up to that. Like, maybe you already answered this, but I was curious if there's a tRNA abundance issue. Like, are they using pretty abundant tRNAs in the particular organism? Or, because that kind of gets to how important and how specific are these natural products?
It's a great question. The first one that we discovered is actually one that used glutamyl tRNA. I didn't talk about that. And that comes from a lactic acid bacterium, Lactococcus. And if you look at what are the most prevalent small molecules in E. coli, it is glutamate. So we thought like, yay. And then we talked to someone who studies Lactococcus lactis, and it's not glutamate, so not yay. And so it doesn't seem to be correlated with the amount of amino acyl tRNA there is. Of course, for the great majority of the organisms where these clusters come from, we actually don't know. There's no metabolomics that has been done that we know what the relative amounts are of the different metabolites. But for the one that we do know, it didn't correlate. So we don't know. We don't know why, and how this evolved. And we can also not predict. We had hoped that the AlphaFold structure would allow us to predict before we do the experiment, because that would make it much easier to find the other 12 that I want to find. And so far, that has not worked out. So AlphaFold is good, but not perfect. And it cannot predict at the moment the substrate selectivity for the amino acid tRNA.
Also one up there.
Thank you for the great talk. I had another question about the Pearl enzymes. Is there something about the structure that limits the enzyme from acting on proteins with long C-terminal tails? Or does it have to be just RIPs and other smaller peptides? And I'm curious if there have been proteins with that modification observed.
Yeah, it's a great question. So as far as we know currently, and we haven't studied all of the 8 that the one that we have studied most puts on a tryptophan. It is quite forgiving with respect to the sequence of the peptide, which surprised us, to be honest. So we don't understand why it's not adding tryptophan to a lot of different things. There are certain amino acids that needs to be spaced a certain distance from the C-terminus, and you need to have those, otherwise it doesn't work. But we've taken the last, I think it was 12 amino acids, and put it on a protein, put it on MBP, put it on eGFP, and then the enzyme does add tryptophan to the protein. So it is a tag that allows you, but then you add an amino acid that, of course, you could have also encoded on the tRNA. On the DNA. But we are very interested in looking at unnatural amino acylated tRNAs that have been used for the codon suppression type of technology, and see if we can tease our enzymes starting to put non-natural amino acids onto proteins. But we haven't yet done that.
If I can add to that, I think at least in eukaryotes, the identity of the C-terminus sometimes determines the stability of the protein. So this could be a really neat tool to tune the stability of proteins.
Yeah, that's a great idea. Yeah, I hadn't thought of that, but yes, you're right.
Laura?
I was going to just ask another mechanistic question. So I know they're not tri-iron proteins, but I'm just going to call them MNIOs or whatever. So those enzymes, you propose for one of them this interesting mechanism where you make that kind of glycol radical later, as opposed to just abstracting that position, which you show doesn't happen.
Yeah.
And I just wondered if you had any calculations or anything like that that might provide insight into why those other steps happen prior to glycol radical.
Yeah, we have not. To be honest, we made that alpha deuterated position because we thought that's where the abstraction. Yes. Also because then I could draw making an episulfide and opening it up, move the sulfur to the alpha carbon. So that was our preferred mechanism. Yeah.
Based on your other
Yeah.
mechanism.
Yeah.
So
Yeah.
that's kind of what I thought you were
Yeah.
doing.
Yeah, so we have a crystal structure, but we don't have a co-crystal structure of that enzyme. So the co-crystal structure I showed you was for Amy and Wei Cheng's enzyme, and so we haven't done any calculations. But yes, we should, probably, because I cannot even tell you what the calculated activation energies are of any of these steps, and maybe some of them are ridiculously high. So it's something that, a lot of work we still need to do.
Okay, with that, let's thank Wilfred for a terrific seminar.